Welcome to the Bodhi Academy. My name is Shrutika and today we are going to continue with our lesson photosynthesis in higher plants. So in our previous parts we have studied what is photosynthesis, where exactly photosynthesis process takes place, and in our part 2 we have covered what is photosynthesis and how the photosynthesis process takes place and how the electron transportation happens during the process of photosynthesis in plants. So in this video today we are going to see what is the meaning of cyclic and non-cyclic photophosphorylation. So cyclic and non-cyclic photophosphorylation. So basically what is cyclic photophosphorylation? So cyclic photophosphorylation is a process wherein it is performed by photosystem 1 independently, and an external source of electron is not required in this process, and it synthesizes only ATP, and it occurs only in the stromal or the integral thylakoid region. Okay, so this is about the cyclic photophosphorylation. So this process, the processes through which ATP is synthesized by the cells in the mitochondria and the chloroplast is called as photophosphorylation. So phosphorylation is basically a process of synthesizing ATPs. Okay, ATP is a form of energy which will be provided to the cells. So the synthesis of these ATPs is the process is called as phosphorylation, and photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in presence of light. So this happens in the process of photosynthesis. So photophosphorylation is the process of synthesis of ATP from ADP and inorganic phosphate in the presence of light. So this is the meaning of photophosphorylation, and phosphorylation is basically synthesis of ATP by the cells in the mitochondria and the chloroplast cells. So this is the meaning of phosphorylation and photophosphorylation. So the cyclic photophosphorylation happens in this way, that is light is accepted by the photosystem 2, that is P700 here, then it will be in the excited state, and then it will reach the electron acceptor, that is photosystem 1, and from there it will go to the electron transport system, and from there again it will be transported to the photosystem 2. So this happens in a cyclic manner. So this is called as a cyclic photophosphorylation process. But what is non-cyclic? So non-cyclic phosphorylation, it is performed by collaboration of both PS1 and PS2 photosystem, that is photosystem 1 and photosystem 2. Both the systems are in will work in the non-cyclic photophosphorylation. So this process requires an external electron donor, and it synthesizes ATP and NADH both. Okay, but in cyclic photophosphorylation only ATP was synthesized, and it occurs in the granule thylakoids only. Okay, the region also changes for non-cyclic phosphorylation. So the non-cyclic phosphorylation involves both the system, that is PS1 and PS2. Over here the light harvesting system will accept the light, then the electrons are in excited state, they will go to the electron acceptor, that will transport the electrons to the electron transport system. From there it will reach the second light harvesting system, from there it will go to the electron acceptor of photosystem 2, and then the electron will be released in form of here the ADP will be changed to ATP, and at the PS2 system the NADP will be converted to NADPH. So in the non-cyclic process you will get ATP and NADPH. Okay, so this is how the electrons are released. Okay, it is not getting cycled. So this is non-cyclic photophosphorylation, and this is cyclic photophosphorylation. So this is C. Over here ATP and NADPH both are synthesized, and over here only ATP is synthesized, and this is for cyclic. It is in a cyclic manner, and this is in a non-cyclic manner as shown in the figure. Now we will move to chemoosmotic hypothesis. So chemoosmotic hypothesis is basically, it is splitting of water molecules occurs inside the thylakoid to produce hydrogen ions and photons. So as electron passes through the photosystems, photons are transported across the membrane because primary acceptors of electron is located towards the outer side of the membrane. Okay, and the NADP reductase enzyme. So over here this enzyme is very important. So here the NADP reductase enzyme, which is located in the stroma side of the membrane, these electrons come out from the acceptor of electron of PS1 photon, are necessary for reduction of NADP to NADP to NADP plus H plus. So over here this is very important, reduction of H plus ions. So these photons are also removed from the stroma. This creates photon gradient across the thylakoid membrane along with pH in the lumen, and gradient is broken down due to movement of photons across the membrane to the stroma through the transmembrane channel of F0 of ATPase, and one part of this enzyme is embedded in the membrane to form transmembrane channel. So the other portion is called as the F1 that protrudes on the outer surface of the thylakoid membrane, which makes the energy packed ATP. This ATP and NADPH produced due to the movement of electron is used immediately to fix CO2 to form sugar, and the product of light reaction used to derive the process leading to synthesis of sugar is called as biosynthetic phase of photosynthesis. So this is how the chemoosmotic hypothesis takes place. So let us understand this process using a figure. So chemoosmotic hypothesis, it is a basic process of ATP synthesis in the chloroplast. So ATP synthesis in the chloroplast takes place through a process called as chemoosmotic hypothesis. So let us see how this mechanism takes place in the chloroplast. Okay, so basically the chloroplast, as we all know, it consists of the thylakoid membrane, that is, this is the stroma region, then there are granum that consist of the thylakoid, which has a thylakoid membrane. So basically this is the membrane, this is the stroma region, so this is the outer region of the chloroplast, that is the stroma region. Then you have the membrane bound granums which are present inside the thylakoid, which are present inside the chloroplast cell organelle. Okay, so this is the membrane. So this pink color thing which you can see is the thylakoid membrane. Okay, so this thylakoid membrane will consist of the PS1, PS2, then this is your electron transport system, and this is your electron transport system, and this is your PS2 system. So basically this process, we know that this is the non-cyclic process, that is, from here what happens is the light energy is accepted by the PS2 system. From here the electrons will move to the electron transport system, that is cytochrome B6F system. From here the electrons will move to the PC, that is for plastocyanin, and from here it will be transported into the P7, that is the PS2 system, that is P700, and here the NADP which is there, it will combine with the hydrogen atom and then it will form NADPH. And this is the ATP synthesis enzyme which is present in the membrane of the thylakoid, which will help in production of ADP, which will combine with the ADP plus P to form your ATP. So this is how the ATP synthesis takes place through chemoosmotic hypothesis. Okay, so this was about chemoosmotic hypothesis. So in this video what we have learned is cyclic and non-cyclic photophosphorylation and how chemoosmotic hypothesis takes place in synthesis of ATP molecules. So thank you for watching today's video. Please don't forget to like, comment, share and subscribe to the Bodhi Academy.